Okay. Well, welcome everyone. I just want to thank everyone so much for being here. It's really valuable to have the connection between researchers and our community. It's very valuable, I think, for everyone involved. I'm just going to ask everyone to mute unless they're speaking. So please go ahead and mute yourself so we don't have background noise if unless you're speaking. And the way we're going to do this is we're going to have a presentation from Dr. Frohlich for about, I don't know, 20, 25 minutes, and then we're going to leave open for questions, as I'm sure there'll be lots of questions. So if you can, hold your questions until the end of the presentation. And I'm going to start with maybe a 5-minute presentation, just talking a little bit about the Foundation's research program and introducing Dr. Rappold and Dr. Frohlich. So going to um, share. Let's see. Let me redo this. How do I go back to Zoom? I think I hit this wrong button. I have to share a specific screen. Okay, I'm not sure. Can you see this screen with this slide?
Yes.
Yeah. Okay. So yeah, it's not doing it the way I want, but that's fine. Okay. So I just want to mention about therapeutic options for FOXP1 or any monogenic neurodevelopmental disorder. The foundation has been doing a lot of research into how we can help um, get there to be treatments for our Fox kids. And there are three broad categories of treatments that are generally seen. So we've been discussing with other, conditions, the foundations and the research programs. There's lots of ones that are kind of established before ours. The conditions were discovered before ours. There are more patients, and the research programs are more established, and we're following their lead in terms of what kind of treatments we should be going for. And the three main categories of treatments that are in the works, just so people understand, are the following. And I should mention before I say this that even for really well-established conditions that have been studied for 30 years and have budgets of $10 million a year, there are very few treatments that are currently available. So this is all very modern science, and we're at a good point in time where science is really flourishing and exploding in these directions. And so even though we kind of have these decades of sort of being behind in research, there's sort of a catch-up period. And even though other conditions, they don't have anything or very few treatments available right now. We expect them to be coming out. They're in trials for some other conditions already, and we can hope that with FOXP1 we can play a catch-up game and get there within just a few years. So I think we're at a very exciting point in time in science, and we're taking advantage of that with our research program. So that being said, the three categories of treatments that we're exploring are AAV gene replacement therapy. I'm going from the bottom up here on this slide, ASO upregulation therapy and small molecule therapy. And as you go up from the bottom, as I explained it, it becomes more intensive treatment to kind of less intensive treatment. So gene therapy, we had an entire call about that in July. So I'm not going to go over it, but we had a research project that we're funding for Dr. Genevieve Konopka and Jay Gibson at UT Southwestern. And that's a very intensive treatment to give gene therapy. It involves, inserting a viral vector into the brain and then trying to produce
Yeah.
FOXP1 from that viral vector. So that's a very intensive treatment, but it's also directly gets at the source, which is the lack of FOXP1 protein that our kids have, or the deficiency in FOXP1 protein that our kids have in their brain, and trying to remedy that directly. ASO upregulation is a little bit in between. It kind of also acts to try to increase the amount of FOXP1 protein that is produced, but it's a different mechanism. And I'm not going to spend time talking about it now. I'll just say that we're currently involved with a company in discussions that's been going on for maybe 6 months now. And we have very regular meetings about trying to develop this research and develop this program. And we're probably will be making a very significant investment from the foundation to develop an ASO upregulation treatment. Um, and it's not as intensive as the gene replacement therapy because it's not a permanent thing that gets, inserted directly in the brain. It's kind of done by intrathecal injection, and it's temporary, so you can kind of wean off it if it's not working for you. So it's somewhat in between, the small molecule and the gene replacement therapy. Now, the top line is small molecule, which basically means a drug. It's a Advil or Tylenol is considered a small molecule, and we're trying to develop a small molecule that may not directly affect FOXP1 production, the production of this protein, but will still benefit our children by kind of regulating downstream effects of FOXP1 deficiency. And so the example of that kind of treatment is what we're going to be hearing about today in this talk by Dr. Frohlich and Dr. Rappold. This is an example of a small molecule treatment, and it's appealing in a lot of ways because it's by far the least intensive of all the kinds of treatments that I've talked about. This is just, a drug that your child might take, and of course, we know that there are possible side effects and risks occurring with any drug, so nothing is 100% risk-free. But relative to the other kind of treatments that I've described, small molecules are something that all of us have taken, many times in our lives, and we're probably very comfortable giving such a thing to our children if they are deemed to be safe. And of course, you can discontinue the taking of a drug at any time if it's not working for you. So that's very exciting, and that's what this project is about, and we're looking forward to hearing about it. So this particular project is about regulating an enzyme whose production is affected by FOXP1 syndrome rather than FOXP1 itself, and hoping that regulation of that enzyme improves the symptoms that our children see cognitively. So I'm not going to, because I've already taken so much time, I'm not going to go into describing these foundation research activities, but all of these projects, whether it's the gene replacement therapy, the ASO treatment, or the small molecule, all of our projects that we're funding, we're also funding clinical projects so that we're in the right place to be conducting trials. You can't just jump into a trial when you have a putative treatment. You have to have done a lot of clinical research about your condition, and we're funding those as well. They all cost a lot of money. So we spend 257,000. I think that might be a little bit low. I think it might be something closer to $270,000 in 2024. We're planning to spend somewhere upward of $350,000 in 2025. And so I just want to end my introduction by making a call for donations, because all this research that we're all hoping to take advantage of and have turned into treatments for our children, we need to do the basic science research, and that's what we're doing now, and it costs a lot of money. And so can you see this web page now? This is the web page for the Rare Disease Day that is coming up at the end of February. We're starting a fundraiser now. You can see we've got a donation of $100 towards a goal of $5,000. So I'm really imploring everyone to try to, I put the link to this in the chat. And if you click on donate, you can see we get one time or monthly. We would love if as many people as possible on this call sign up for monthly donations, whether it's $50 or $100, or if you can afford it, $250 a month. That would be really fantastic, because that would get us to the point where we can actually make these investments in these research projects. The $270,000 or $250,000 of research grants we made in 2024 were largely funded by three families. So my family gave about over 150,000 of that ourselves, and the Targon family had a big fundraiser, and the Whitney family raised a lot of money. But we only basically have three families that are giving $10,000 or more. And relative to the size of our community, we know that that's small. Other organizations that have similar sizes or a little bit bigger are raising much more money than we are. So we're really asking people, if it appeals to you, the research that we're describing today, the possibility of treatments to come to our kids within a few years, please donate to make this a possibility, because I think the lead families, only have so much bandwidth, and it can only last for so long before we kind of are running out, and we don't want to lose steam in these research programs. So please, please, please contribute. We know a lot of you took part in the races, and that was basically the complement that, of what the individual families gave. And we need to really beef up our broad base of fundraising, where we have, say, many monthly donors to make projects like this possible. So with no further ado, let me now introduce Dr. Frohlich and Dr. Rappold as they describe their research project that we're very happy to be funding from the foundation into a PDE10A inhibitor. Professor Rappold is the senior professor at the Institute of Human Genetics at Heidelberg University Hospital. She studied biology at Stanford University and the universities of Konstanz and Heidelberg, completing her PhD in cytogenics and molecular genetics at Heidelberg. Following postdoctoral research at institutions including the MRC in Edinburgh and EMBL in Heidelberg, she became the director of the Department of Molecular Human Genetics at Heidelberg University in 2002. Professor Rappold's research has significantly advanced the understanding of the genetic basis of neurodevelopmental disorders, with a particular focus on the FOXP1 gene. She has been appointed to a senior professorship at the Faculty of Medicine at the University of Heidelberg. Okay, Dr. Henning Frohlich is a research group leader at the Institute of Human Genetics at Heidelberg University Hospital. He leads the research group Mouse Models for Neurodevelopmental Disorders, focusing on understanding the genetic and molecular underpinnings of conditions such as autism spectrum disorders and intellectual disabilities. His work involves developing and studying mouse models to investigate the role of genes like FOXP1 in neurodevelopment. Professor Frohlich and Professor Rappold have co-authored several publications exploring the impact of mitochondrial dysfunction and oxidative stress on cognitive and motor impairments in FOXP1 syndrome. Together, they have collaborated on research projects investigating the potential benefits of a PDE10A inhibitor for individuals with FOXP1 syndrome, aiming to develop targeted therapies for the syndrome. And so with that, I'd like to turn it over to Dr. Frohlich.
Okay, thank you very much, Samit, for the kind introduction.
Yes.
So
let me make sure that everyone can share. Can you share? All participants can share.
It seems to be possible.
Yes, go ahead.
So can everyone see my screen?
Yes.
Okay. So again, thank you very much for the kind introduction, and I would like to welcome you all. And I'm very pleased today to have the opportunity to provide you with some background information on our research topic, on our current research topic, which is funded by the FOXP1 Foundation and which aims to investigate a potential new therapeutic target of FOXP1 syndrome, as Samit just explained. So for me as a scientist, meetings like today's show once again what the real goal of basic medical research really is. And this is to gain a deeper understanding of the underlying pathomechanisms of a disorder, which can enable the development of a specific and effective treatment to help those affected. But before I go into detail about the actual project, I would like to present the findings on which our project is based and why we believe that our new treatment strategy could significantly alleviate at least some of the deficits that we observe in FOXP1 syndrome. So I'm sure you're all very familiar with the symptoms, so I don't need to go into any more detail. However, I just want to mention that our research at the Institute focuses primarily on the altered neurodevelopment and brain function in FOXP1 syndrome and the resulting behavioral and cognitive deficits that we see, as well as the motor disorders. So research into the causes of a genetic disease, of course, cannot, be carried out on humans, and certainly not on those affected. And for this reason, we often use model organisms, such as the mouse. So one big advantage of a mouse model is that it has a high genetic similarity, but also many behavioral patterns that we find in the mouse are similar to humans. And for our studies, we use the FOXP1 plus minus mouse, which, like individuals with FOXP1 syndrome, has a loss of one FOXP1 allele body-wide. But also some behavioral deficits can be found in these animals that are similar to what we know from patients. So on the one hand, these animals show reduced neonatal ultrasonic vocalization. To explain that in brief, if you separate neonate mice from their mother, from the dam, then they start calling. And this calling behavior is interpreted as a kind of early social behavior. And in addition to that, this calling or the mechanisms that control the calling are very similar or almost exactly the same to those that control speech and language in humans. In addition to that, these animals exhibit hyperactivity, they show increased anxiety, but they also have deficits in learning and memory. And in the last years, our research, but also the research from other research groups, for example, the group of Professor Konopka showed that one brain region in the forebrain is particularly affected in FOXP1 haploinsufficiency. That means the cause that one copy is lost, and this leads to FOXP1 syndrome. And this region is the striatum. And because of that, our main focus in the last years was on the striatum and striatal function. And what we found was, for example, Samit already mentioned that the neurons in the striatum have a mitochondrial dysfunction. So the mitochondria are cell organelles that are the power plants of the cell, and they are used to generate energy. And of course, if energy production is somehow altered, this may impact on the neuronal function. And what is quite clear is that for the most part, the symptoms that we see in our mouse model, but also in patients, can be attributed to striatal dysfunction. So this picture now shows you, on the one hand, the human brain with the striatum, which is marked in color. And on the right, you see the mouse brain, and different brain areas are colored. And in bright blue, you see the striatum. And as I already mentioned, we focus on the striatum. We, of course, investigated the gene expression in the animals that have FOXP1 haploinsufficiency. FOXP1 is a transcription factor. That means this gene codes for a protein that regulates a huge bundle of different genes. And we analyzed the gene expression in the FOXP1 striatum. And of course, we found many genes that show an altered expression, but one of these genes is particularly interesting. And this is the so-called PDE10A. And we found a 50% reduction of this gene to be expressed in the striatum. And now I will explain why this gene is so interesting for us. And it might be important in the treatment of FOXP1 syndrome. PDE10A codes for an enzyme. It has a very complicated name, which I show you above. This is not important, but the important point is that PDE10A, this enzyme, eliminates two molecules, which are cAMP and cGMP. And these two molecules, which we call which are so-called second messengers, regulate a lot of important cell functions. And in neurons, we know that cAMP and cGMP increase the neuronal activity. So as PDE10A, this enzyme eliminates these two molecules, it reduces neuronal activity. The second part, which makes PDE10A so interesting, is that it is almost exclusively expressed in the striatum, but not in other brain areas. So here in this left picture, you see one hemisphere of the mouse brain in which PDE10A mRNA, the messenger RNA, which in the end then is translated into PDE10A protein, is stained. And this dark staining here shows you the striatum. And on the right, you see the protein, which we marked with a specific antibody, and it's shown here in green. And due to its expression, that it is exclusively expressed in the striatum, and due to its function, that it reduces neuronal activity, PDE10A in the last 10 years became a very important therapeutic target for several disorders in which the basal ganglia are involved. And the striatum, just to explain that, is one of the major parts of the basal ganglia. So this holds true, for example, for neurodegenerative disorders such as Parkinson's disease, but also Huntington, but also psychiatric conditions like schizophrenia, autism spectrum disorder, Tourette syndrome, and addiction. And I will now explain you why we believe that PDE10A might be a very good target to treat FOXP1 syndrome. And with this, I have to go a little bit deeper into basal ganglia circuitry. So this is very complicated, and I try to make it as simple as possible. The striatum has two main cell populations, and these are, on the one hand, the D1 medium spiny neurons and the D2. And at first, I will explain to you what the D1 medium spiny neurons do. So if these neurons are activated, the direct signaling pathway is active, and this direct pathway, which in the end activates parts of the cortex, is needed for the initiation and execution of voluntary movements, but it also controls certain behaviors, such as, for example, activity and anxiety. And there is this second population of neurons which belong to the indirect pathway. If these neurons are activated, then this prevents unwanted muscle contractions, and it also prevents certain behaviors such as increased anxiety or, for example, hyperactivity. And what we know is that the balance between these two pathways is very important. And if this balance is disrupted, as you see in several disorders, then you will find behavioral deficits. And interestingly, in the last 10 years, it became more and more clear that PDE10A plays an important, a crucial role in regulating the direct and indirect pathway. And what we know is if we block this enzyme, the PDE10A, with an antagonist, then we mainly hit the D2 medium spiny neurons, and we mainly activate this population of neurons, and this leads to an upregulation of the indirect pathway activity. And with this, I will come back to all these disorders in which the basal ganglia, in which the striatum shows a disruption. What we know from many different experiments that were performed in animal models, but also in humans, is that all these disorders show a disruption of the balance of the direct and indirect pathway. So it is believed that also in autism spectrum disorder, the direct pathway is overactive, whereas the indirect pathway, which inhibits certain behaviors, is not active enough. And what is quite clear is that by targeting this enzyme PDE10A and by blocking it, we can increase the activity of the indirect pathway, which may then lead to the balance of the system. And this can, in the end, impact on behavior, and it can improve behavioral deficits, but also motor dysfunction. And several years ago, when we found that PDE10A is downregulated in our FOXP1 plus minus mice, which reflect FOXP1 disorder in humans, we had the hypothesis that a continuous inhibition of this enzyme of PDE10A from birth will improve the existing behavioral deficits that we find in our animals. And at that time, we decided to use a drug to inhibit PDE10A, which is called MP10. MP10 is one of the very new drugs that were developed. It is very specific, so we only hit PDE10A and not other phosphodiesterase that exist in the brain. This is really important. And it showed low side effects in animal models, and it showed low side effects also in humans in clinical studies. And this is now what we did. So we decided to treat our animals and to test behavior. And we started our treatment directly after birth in pups, and we treated the animals for a time period of around 30 days. And in between this time period, we performed several behavioral experiments, and we focused on early social behavior, we focused on hyperactivity and anxiety. And I can go into more detail if this is wanted afterwards, after the talk, if you want to get more information how these behavioral experiments are performed. And this is now the outcome of our treatment. So I told you at the beginning, our animals show reduced ultrasonic vocalization, they have hyperactivity and increased anxiety. When we treated the animals with MP10, ultrasonic vocalization was normalized. These animals show a normal call number compared to wild-type littermates. When you look at hyperactivity, it was not present anymore in animals that were administered with MP10. And this also holds true for anxiety. Increased anxiety was gone, and they showed normal anxiety compared to wild-type animals. And for this reason, it was quite clear that MP10 treatment completely corrected the behavioral deficits we looked at in FOXP1 plus minus animals. But with this, I want to come back to what I told you at the beginning, that we can never investigate the underlying reasons of a certain disorder in humans, and therefore we use models. And the last years, we used our FOXP1 plus minus model. Of course, a mouse has a lot of advantages. So for example, we can investigate the underlying molecular alterations. But in addition, of course, we can also check behavior and physiological aspects. But one should keep in mind a mouse is always a mouse. And although we are closely related, as we are both mammals, there are differences. And therefore, it has to be tested whether the molecular deficits that we find in our mouse also exist in human cells with FOXP1 deficiency. And that was the reason why we thought of using human cells and why we thought of human striatal organoids to investigate whether some of the, or at least some of the molecular deficits that we see that seem to be the underlying causes of the disorder can be found in human cells as well. Now, of course, it's not clear to everyone what an organoid is. So therefore, I want to explain it in brief. An organoid is a tiny self-organized three-dimensional tissue culture that is derived from, in most cases, from human-induced pluripotent stem cells. And these cultures can be crafted to replicate much of the complexity of an organ, and in some cases to express selected aspects. So for example, if we want to have a look at a certain cell type within an organ. So this picture below now shows you how these organoids are generated. At first, we need cells from a human being. Then these cells are reprogrammed into human-induced pluripotent stem cells. That means these cells can be differentiated in all kinds of cells that we find in the human body. And in our case, we want to differentiate these cells into striatal cells and generate a striatal organoid. And we will do that with patient cells with FOXP1 deficiency, and we will compare them to control cells, to iPS cells that we generated, that we got from healthy individuals. And we have 4 different cell lines that we will now investigate that were kindly provided by the Buxbaum Laboratory in the Sievers Center. 2 of them are from individuals with FOXP1 syndrome. So one patient carries a missense variant, so we have an amino acid change within the FOXP1 protein, and the other one is a deletion, so one part of the FOXP1 sequence is disrupted. And the controls are cells from siblings. This is just an example how such organoids look like. I have to mention these organoids are generated by one of our collaborators, Julia Ladewig, who is a group leader in Mannheim. So Mannheim is very close to Heidelberg, and we will closely work together. And Julia is an expert in the generation of brain organoids of all kinds, also of striatal organoids. So, for example, on the left you see a whole forebrain organoid, and on the right you have a striatal organoid. And just to explain that, the different colors are, on the one hand, markers for different proteins, which tell us which kind of cells we're looking at. And with DAPI, so this blue staining, we marked the nuclei.
Okay,
So
well,
now,
I'll text you when when that comes in.
Little car, or can I just write on the box?
so the questions that we want to answer by using these organoids is, at first, are there morphological differences visible between the patient cells and the patient organoids compared to the control cells and organoids? For example, in our mouse model, when we investigated the striatal neurons, we observed morphological differences. And of course, that also impacts on function. So on the left here, you see a striatal neuron from a wild-type mouse with this long dendritic arbors. And when you compare it to this mouse with FOXP1 deficiency, you can see that the structure looks different. So this is one aspect of our work. But another aspect, and this is very important, is do we also see a reduction of PDE10A expression in the medium spiny neurons in patient cells? And another question to go into more detail is, are there expression differences between these two subpopulations of neurons that we find in the striatum? The ones, the D1 medium spiny neurons that belong to the direct pathway, and the one that belong to the indirect pathway. And what we will also do is that we have a look at the gene expression in general in the medium spiny neurons and compare it to the control cells. And furthermore, at the beginning, I told you that we found a mitochondrial dysfunction in the striatal neurons in our mouse model. And of course, this raises the question, does a mitochondrial dysfunction is also present in patient cells? And now I will come to the most important part, of course, of our work, the treatment with MP10. So what we will do, of course, is treating these organoids with our PDE10A antagonist, and then we will investigate the effect, on the one hand, on organoid and cell morphology, whether it ameliorates, for example, alterations that we find, and how it impacts on gene expression in the dopamine, in the D1 medium spiny neurons and in the D2 medium spiny neurons. And another aspect, of course, is if we find a mitochondrial dysfunction in the patient cells, like the mitochondrial dysfunction that we found in our mouse model, how does our treatment impact on mitochondrial function? Does it, what we hope is that, of course, it ameliorates the mitochondrial dysfunction. And from the literature, we know that MP10 indeed impacts on mitochondrial function. Another aspect of our work is not done in the organoids, and this is an aspect that we will investigate again in our mouse model, and this is the question, when is PDE10A expressed in the developing striatum? So we know that it is present at late embryonic stages, but most of the stages that we investigated were postnatal time points. And for this reason, we will investigate the presence of PDE10A in striatal tissue at different time points of mouse development. And the moment we know at which time point it occurs, then we will compare that with the embryonic and fetal stage, because there are ways to correlate mouse and human development. So the overall aim of our study is of course, to investigate and to find out whether the findings from our mouse model are transferable to FOXP1 syndrome, are transferable to humans. And of course, another very important point is, how do patient cells react to MP10 administration? Is the treatment beneficial? Does it ameliorate some of the molecular deficits that we observe? So in the end, our treatment shall lead to a better assessment of the MP10 value of the treatment in FOXP1 syndrome. And this brings me now to the end of my presentation, and I would like to say a big thank you to the FOXP1 Foundation and of course to you, dear parents. Without your financial support, this organoid project would not have been possible. And with this, thank you very much for your attention, and I will be very happy to answer any questions you may have.
All right. Thank you very much, Dr. Frohlich, for that wonderful presentation. Everyone is clapping, though they're muted, so you can't hear it. But that was very clear. So we're going to open the door now to comments or questions. I just want to say something very quickly, which is, I'm sure a natural question people are having is a timeline. That is, like, when we might expect this research to turn into a treatment or a drug we can actually give our children. And it's very hard to pinpoint that. It really depends on the science. It depends on the result of this experiment. So it's really hard to promise anything. An important point, which is unfortunate, which is that this drug is not FDA approved or approved by the regulatory agency in any country. So it's not available to give to any individuals for any condition, for any reason right now. It's only available as a research tool, as a research drug, and that's, how Dr. Frohlich and Dr. Rappold have gotten a hold of it. But the good news is it is being developed for other conditions by various companies, and those projects are ongoing. And if it gets approved by the FDA for another condition, that really speeds up our process. So we can hope that that happens and we can continue the research. So right now, we're not at the stage where we can go into trials yet anyways. That's the purpose of this project. So I should mention that obviously it's scientifically important to understand that this drug works on human cells and not just on mice, but also that's basically a requirement or a necessity of the regulatory agencies as well. So we can't go into trials unless we demonstrate the efficacy of this at fixing some phenotype in human cells, such as an organoid. So that's a reason we're doing this, is to lay down the science so that the FDA will let us go into trials. Whether or not we go into trials or how we go into trials will vary greatly depending on what stage the drug is in development for other conditions. So we're going to wait and see, but 6 months, 1 year, when this research is finished, what our plan will be determined largely by where the drug is for other conditions. So we're in a little bit of a wait and see, but we can definitely hope that we're in trials within a couple of years. That's definitely an ambitious goal, but something that is realistic. And once anyone who's in the trial can see benefits for their kid right away, hopefully. So well, with that being said, let me now open the floor. If anyone has questions, you can raise your hand and I'll call on you. There's a lot of us in the call, so please use the hand raise function on Zoom, and I'll just call on people as I see hands raised. Go ahead, Eddie.
Thanks for setting this up. Doctor, I think I'm going to ask what a lot of the parents are going to want to ask is there a hypothesis on how effective this treatment might be as the child starts to age, right? It sounds like there's going to be much more impact if it happens very early, like a baby's just born. But if your child is, 5, 10, 20, is our hypothesis, how effective it will be to kind of reverse what they've kind of expressed throughout their life.
Yeah. So this is something that we don't know yet. So there is a lot of investigations that we do concerning this aspect. So what we do at the moment, just to explain it, is we do treatment trials in which we treat the animals at different time points of development. So, for example, we treat the animals at a postnatal stage, which reflects the age of 1 year. We treat the animals at a stage which reflects the age of 3 years. And in addition to that, we also treat animals at adult stage to see whether this ameliorates the deficits. At the moment, there is not enough data available yet to finally conclude. So therefore, I don't want to guess what is really the case. So at the moment, I have the feeling we should start at an earlier stage, but whether starting directly after birth is really necessary, this is the big question. But there are a lot of trials that we are performing. So I have 2 PhDs that work on that. This is investigated in animals. And another trial that we perform, just to mention that, is we treat the animals for a short time period after birth, and then we stop the treatment because it might be this that the treatment impacts on brain development, and that you have a certain time period which is crucial. As you said, that it's really crucial to start at an early age, but the question is whether we have to go on with the treatment. And what we do is we treat the animals at a young stage, and then afterwards, if these animals are aged, we test them again, whether the behavioral deficits are still there or whether this early treatment, which we then stopped, was already beneficial.
Yeah, so let me add to that a bit. So, Dr. Frohlich, as the researcher on the study is being very cautious. Of course, we would not be doing, this study if we were not optimistic that this would be able to treat all of our children of all ages. And I can say we can draw inspiration and hope from treatments for other conditions. So, you may have heard that there's another condition called Fragile X, which is another neurodevelopmental disease, and they're testing a very similar enzyme inhibitor called a PDE4D inhibitor. It's a different enzyme in the brain, and they're already in clinical trials for that. And the results are not out, but they're seeing positive results in adults even. So um, we can very much hope that there's no reason why, we couldn't see similar results for our kids. So we're talking about adults being helped by this. And of course, yeah, the range of how individuals might be benefited by this um, will vary based on particular mutation, based on age, based on a variety of factors that we don't necessarily have control over. But definitely, we hope that this will help all of our children. Um, I want to mention one other thing I was asked in the chat. Someone said, looked like Mark is testing this for schizophrenia. We actually did have a call with Mark, with Dr. Frohlich and Dr. Rappold and Carl and I. And they basically informed us they wanted to see more evidence of benefit of this drug for individuals with FOXP1 syndrome. And so we're going to continue that conversation. But yeah, thank you for posting that. And we are on it. There was a question from Lisa. She had her hand raised, but I'm wondering if it was answered because the question in the chat seems similar to what we've just been discussing. Lisa?
Yeah, thanks. It was. I appreciate that information.
Any other hands raised? Any other questions? So Claire asks in the chat, are other organizations in the clinical trial phase? And the answer is yes. Merck is in the clinical trial phase for treatment of schizophrenia. Noema Pharmaceuticals is in the clinical trial phase. They're actually further along than Merck for treatment of Tourette syndrome, which is great news. These drugs could be, approved for other conditions if everything goes well with the trial. That's a big question, of course, but could be approved within a year, year and a half, 2 years, which gives us good, timing for us to get involved as we see how that's developing. So we're going to definitely keep a close eye on what's happening in other companies. Catherine asks, have there been any side effects in the mice? I'll pose that to Dr. Frohlich and Dr. Rappold.
No, there were no side effects visible. So we didn't observe any behavioral or motor deficits.
Okay, I'll come back to Lisa in a second. She has her hand raised, but there's another question. Once they are approved, then there is potential for off-label use. Yes. So, um, once a drug is approved for another condition, if you in consultation with your doctor, feel that it would benefit your child, you can get off-label use, even if it's not approved for your child's age range.
Got it.
But the foundation would not be involved in recommending that until we have done our own due diligence on testing the safety and efficacy of this drug for FOXP1 syndrome, but individual parents can make the decisions as they feel with their own doctors once it is approved for another condition. And of course, that would not be covered by your insurance here in the US. I don't know rules in other countries, but if you use it off label, that would be something you would pay out of pocket for. All right, now we'll go to Lisa, who has her hand raised.
Yeah, thank you. I am brand new to this diagnosis for my son, so
Here,
forgive
lie
me.
down. I can do it.
Forgive me if this is old, a subtle question for lots of
Just
other
make
parents
sure I'm
and
on mute.
caregivers, but I'm wondering, with all that you're learning about the impact of this mutation on brain development and function, do we know anything while we're sort of waiting for this research to move along about things that we can do now that just support this area of brain function? Are there certain vitamins and supplements or existing easy access medications that at least theoretically should help this area of the brain function properly?
Shall I comment on that?
Yes, please.
So, of course, I'm not a medical doctor, so I'm very careful in. I wouldn't suggest anything which is not approved by a doctor. But I think so far there is no medication known that really helps. Of course, there are treatments that are commonly used in certain deficits that we find in ASD. There are medications that target anxiety or OCD and things like that, like aripiprazole and risperidone, for example, which are known, or Prozac. Some children might take Prozac against the anxiety. However, apart from that, I don't know any drug which might be used. But as I said, this has to be, of course, the better is to ask a doctor whether there are certain other drugs that can be used to ameliorate certain deficits.
Yeah, I'll also just follow up and just concur that there's no blanket medicine or treatment that we know in terms of like a drug or something that or vitamin that we could take that we recommend for all patients. Individuals have different symptoms, and so we ask, patients to consult with their doctor. Often the treatments that people are getting are treatments for particular symptoms. For example, anti-anxiety medication is pretty common in our community. I would recommend that you join the Facebook page that we have if you're not already on. And if there are particular symptoms that you are curious about for your child, you can post about them and see what other parents are doing. And I think that's a good way that parents have shared treatments that their doctors have recommended for their own child. But as of now, there's no blanket medication that we recommend for all of our children. And that's like kind of what we're trying to develop here. I'm going to try to address some of the other questions that have shown up in the chat. Um, is there a way to test for the syndrome at birth? Absolutely. You can get whole exome sequencing even in utero. You can get so certainly at birth you can get whole exome sequencing. So it is possible to test that for birth. And of course, we'd love it if whole exome sequencing genetic screening was done at an earlier age, and that's something that we are advocating for and others are advocating for. Have all the FOXP1 variants been identified? So that's a little bit of a tricky question to address. So basically, FOXP1 is a large gene. There's 2,034 nucleotides, and you could have a mutation or a deletion of any one of those, and that would cause some change, and it's considered FOXP1 syndrome if it's pathogenic. And which mutations are pathogenic, which are not. It's not 100% been verified, but it's basically verified by someone getting the mutation and then displaying the symptoms, and then that particular mutation becomes labeled as pathogenic. So if somebody else gets the same mutation, then we can say that, yes, you're likely to have symptoms. Let me see.
So we cannot really conclude yet if all the variants, the pathological variants, have been detected so far. And Samit, I'm really impressed about your knowledge on genetics by now, which you have gained in the last year.
Yeah, so I got my son's diagnosis in 2023, and I'm not a doctor by training. I'm a mathematics professor, but I spend most of my time these days studying FOXP1.
Samit,
Okay.
I just saw there was a question concerning the animals, whether they have any changes in diet or weight gain compared to wild type animals. So I can
Yes.
comment on that.
And
This
actually, there was another question that you can come at the same time, which is, have the mice seen any increase in intellectual abilities with treatment by MP10? So maybe you can comment on both of those items.
Okay, yeah, I will comment on both. So this is an interesting question, and this is something that we're testing at the moment. So in our treatment trial that we performed at the beginning, and which I presented to you, we didn't have a closer look at cognitive dysfunctions. And at that time, we didn't even know, when we did this study, that they have cognitive alterations compared to wild type animals. And now that we know, we indeed test that. We test different time points of treatment, as I already explained, and different time periods. And we do behavioral experiments to investigate whether the cognitive functions are ameliorated by the treatment or not. But there we don't have enough data yet to conclude. The second point was whether these animals show differences in weight gain after treatment. And that was not the case, at least in our first study. And in the second that my PhD student now performed, we didn't see any change in weight. And this may be a big advantage to treatments that ameliorate some of the symptoms, like risperidone or aripiprazole. These antipsychotics have side effects that include excessive weight gain, because in this case also cells of the hypothalamus, for example, are targeted. And this may be a real big problem, that if you have a longer time period of treatment, that this increases weight and may lead in the end to diabetes. And at least from what we know, this does not seem to be the case with blocking PDE10A, at least with MP10.
Can I follow up a bit on the intellectual abilities? So if we believe that this is going to improve, neural development in the striatum and neural connectivity in the striatum, then it stands to reason that this could improve speech and language development. That's obviously an optimistic goal for this. And you saw improvements in neonatal ultrasonic vocalization in the mice, and you could hope that that parallels communication and speech in humans. So isn't there some optimism that this drug could improve at least communication and speech and language development in individuals?
Well, yes, indeed. Of course, one has to be a little bit careful, because ultrasonic vocalization is not speech and or language in mice, because this is a reflexive behavior, which can be compared to the crying of a baby. So it's not learned. Because at the time point when you investigate ultrasonic vocalization, these animals are completely deaf. They don't hear anything. So that's a difference compared to humans. Humans already in the uterus, at fetal stages, they can hear. This is not the case in mice. So it's completely clear this is not comparable to speech and language. But I mentioned that already in the talk. The mechanisms that control sound production in mice are the exactly same mechanisms that control speech and language. And therefore, I'm optimistic that it may improve speech and language in humans. Yes, this is the case.
Is there anything that parallels speech and language in mice that are comparable to humans of adolescents or adults? And then can you test that, or have you tested that?
No, in this case, mouse is not a good model. So speech is something very special that almost exclusively can be found in humans. Of course, we also have some other animals like dolphins that have a kind of speech, but this is something that we cannot test in rodent models. To some extent, you can investigate learned sound production in zebrafinch, for example. There is some research on that. And also in zebrafinch, there is FOXP1. So there are trials to investigate FOXP1 function in birds and zebrafinches. Yeah, the mouse, as I said, is not a good model to investigate speech and language. This is not possible.
Okay, let me address some of the more questions in the chat. They're about trials. One is, will the foundation facilitate clinical trials? And the other question about trials is, how will you choose patients for clinical trials? So yeah, we're not there yet, is the short answer to this question. It really depends on the shape that it takes. So what the stages of this drug for other companies, what company we ended up working with. We will certainly assist clinical trials in the way that we have this connection with parents. We have the ability to communicate with all of you, and that'll be very valuable for any company that wants to develop this drug. One thing I can mention is a little bit of a preview. We're going to roll out a relationship with Citizen Health, which is a company that provides retrospective natural history studies. So it's going to basically be documenting our kids. This will be for US-based patients, and we're going to be rolling this out in the next month or two. And this will be very valuable, and if we can get as many volunteers to this as possible, and it's just basically a 5-minute sign up, and that's all you have to do, and then there's no more work for the patients. But what it enables us to do is to collect data that we can then use. We collect this data, and that makes it appealing for the companies that, oh, you have all this data on patients that we can use in the clinical trials. And so we will be involved in those ways with the clinical trials. Financially, if we're going to be involved in clinical trials, again, it's too early to say. It depends on how this looks and what stage of development. In terms of choosing patients for clinical trials, that's really going to be up to the companies that are running this. And yes, we'll accepting volunteers, and people can volunteer for clinical trials when the time comes. But again, this is, a couple of years down the line at least. So we're not really ready to answer these questions. Another question is about environmental factors into the causation of FOXP1, as this is largely due to de novo mutations. No, there hasn't been any research into that as far as I know. 3 cases from one area is not particularly large necessarily. This is usually a function of the availability of medical diagnoses and, how likely your doctor is to recommend whole exome sequencing. So in other areas, they just don't recommend it as often, and that's why there are fewer cases. For example, if you're in a major medical location like Indianapolis, or here in the Triangle area of North Carolina, where we have Duke and UNC medical schools, we have a lot of cases here, primarily because the doctors are very good at recognizing genetic conditions and recommending whole exome sequencing. That's really what it is. I'll just mention, people ask about how this happens. Now, every individual has mutations in their DNA. Roughly estimates are roughly 60 to 100 mutations you might have in your DNA. Now, this might happen in a gene that either, you don't notice what it does, or it causes something that doesn't cause problems. Like maybe it changes how you taste something, it changes your height, changes your hair color. Unfortunately, FOXP1 is such an important gene in your body. It's a transcription factor that's involved in regulating many other genes and many other production of many other proteins. It gets involved in everything. And so it's unfortunate for our kids that their mutations happen in this particular gene, but there's no reason to think that there are environmental factors that are particular to FOXP1 other than, in general, environmental factors increasing likelihood of genetic mutations such as parental age or exposure to toxicity. But that's just generally causing, believed to perhaps increase the number of mutations, but that's all marginal. It's all within a range. Everyone has mutations. Some people might have slightly more than others. More messages.
Well, as I see, it was asked how many animals we tested and how many animals benefited from that. So I can't give you a percentage, but so we always, in behavioral studies or in treatment studies, we have to use a high number. So we have to do a power calculation at the beginning, and how many animals we need to answer a certain question. So we tested around 20 animals per group, and almost all of the animals that had this FOXP1 deficiency that were treated showed a beneficial effect. So it was highly significant, the outcome.
In the second half of that question was, this is a treatment that is given once or twice or ongoing forever, and I believe this is a treatment that would be applied regularly, perhaps daily, right?
Yes, at least in the mouse, we applied it or we administered it every day. However, whether this is really necessary is something that we don't know. This, of course, depends on the half-life of the drug, and this might be different in humans. So there were also other trials with PDE10A inhibitors in rodent models, but also in humans there were trials, and there it was not administered daily, but every 3 or 4 days or something like that. And this, of course, is also dose dependent.
Yep. Dosage and frequency of treatment is something we can draw largely from other trials that are ongoing, and it'll probably vary by age range. So we'll probably get a lot of information, especially as this ties into safety, because there will be certain dosages that are proved to be safe or demonstrated to be safe in other conditions. And so that will largely guide how we go in terms of dosage moving forward. Again, that's a little bit early at this stage. We're just trying to prove the efficacy and, of this drug. So I think we've exhausted our questions. Is there anyone else with a hand up or a question in the chat? I don't think there's anything we missed, but if there is, please let me know. Okay. If not, I just want to thank you all once again for joining this. It's really valuable to have the buy-in from the community and for our researchers to meet our parents and know what their research is for. We are all so thankful, Dr. Frohlich and Dr. Rappold, for the work you're doing and trying to improve the lives of our children. We really appreciate it and are proud to support your research.
Yes, and maybe I can also add that we really do hope very much that this treatment may be beneficial. It just takes a little bit more time to find it out.
Thank you so much. And I'll just finally, end with just I mentioned this earlier, but yeah, please stay connected, all of you, to our Facebook page and watch for posts there. As I mentioned, we're going to have a few rollouts in a few months. We have the Citizen Health relationship, which is basically a sign up that you do. They get access to your child's medical records, and then they're able to use that data for use in the clinical trials that we might have for this condition, for this treatment, or any other treatments going forward. And I'm sure there's going to be a lot of questions people have about privacy and of that data and whatnot. And we'll be sure to address those questions when we roll this out to make sure everyone is comfortable with what it is we're asking people to do. But just please keep a lookout for that. I'll mention we have another research project that we're funding for Dr. Paige Seiper at Seaver Institute, and we're going to have a call very similar to this again within the next month or two. This is really going to be important for patients and parents to be involved in, because this is a clinical study. So we're going to be enrolling patients in that study. You get good information about your kid if you enroll in that study, but moreover, this is data that's going to be collected to generate a scale that the FDA approves as a scale that you can use in clinical trials. So we're trying to set the stage for having clinical trials for this drug or any other treatment that we come up with in the next few years. We want to make sure we're ready to go to the FDA. You have to develop some FDA approved scales to evaluate the effect on children and adults. And so we're developing those now. And so we need to enroll patients for that trial. So that's going to be coming in the next month or two. It'll be announced on Facebook, just like this was. So please keep a lookout for that, as well as for the Citizen Health sign up. So two really important things that are going to be announced in the next couple months on our Facebook page. So please keep a lookout for that. And thank you all for joining this call. Bye, everyone.
Thank you very much. Bye.